All right, everybody. So today we're going to talk about the CRISPR technique. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. So it's quite a mouthful, which is why we refer to it as the CRISPR technique. Now, you've all heard a lot about this in the media. So I'm just going to cover the details about how the CRISPR system works so that it makes more sense as we talk about its applications in class. So, the cool thing about CRISPR is that it's actually the bacteria's way of having an immune system. It's referred to as an RNA guided defense mechanism. So, if you think about our adaptive immune system, where we have B and T cells that attack invading organisms that come into our body, this is what the bacteria is doing with the CRISPR Cas system. So, the kinds of things that infect bacteria are viruses, such as a bacteriophage. And so, maybe you've seen a picture like this before, where the bacteriophage is this creature right here. It inserts its DNA into an organism, in this case, a bacteria. And what the bacteria then does is it actually takes the DNA of the phage and it incorporates it into its own genome. So the bacteriophage infects the cell and the DNA is inserted, and the bacterial cell captures that DNA and sticks it into its own genome. So these light blue sequences here are DNA sequence, and the repeat sequences are also DNA sequences of the bacteria. And what's going to happen is that the pieces of phage DNA are going to get chopped up and put into this region. And then right nearby this CRISPR array within the genome of the bacteria is another region called the Cas region. And these are several different genes that are made into proteins. And these proteins are then responsible for cleavage. They primarily are code for enzymes of some sort. So, as I said before, the invading phage DNA is cut into pieces by the Cas proteins, and it's inserted into the CRISPR array. So, in this case, the little diamonds are actually the bacterial DNA, so the host DNA, and then these little rectangle pieces are different pieces of phage DNA that have been incorporated into the bacterial genome. So, what happens next is that whole sequence now that includes the phage DNA and the bacterial DNA is transcribed into a really long piece of messenger RNA. The long piece of messenger RNA is then cleaved into smaller pieces. These smaller pieces are called cRNAs or CRISPR RNAs. So why would the whole system work this way? The idea is that the bacteria wants to be able to recognize the phage DNA and degrade it. And so it's now generated pieces of complementary RNA from the phage DNA. And now these complementary pieces of RNA can actually bind to the phage DNA. And so here's this sequence right here, this piece of RNA. It binds exactly to a region within the phage DNA, and that targets the Cas protein to also bind to this region. And when the Cas protein binds to the region that's been identified by the CRISPR RNA, this is now a complementary base pairing situation here, and the Cas protein's job is to cleave double-stranded pieces of DNA. And so when the Cas protein binds, it cuts the phage DNA, both strands of the phage DNA. This then leads to destruction of that DNA because the DNA has been now chopped up into little pieces and the virus can't reassemble. So the bacteria is basically able to get rid of the phage DNA before more viruses can be made. So that's how it targets for destruction the interfering, the DNA that's come in from the virus. Okay, so that's the way it works in bacteria. But what's really amazing about this system is that scientists have discovered a way to modify this system so that you can use it in a eukaryotic scenario. So now imagine that you have some eukaryotic cells in a dish, like stem cells or any kind of human cells, really. And your goal is now, rather than using the system to target a viral DNA for destruction, we're going to use the system to target a particular region of the eukaryotic genome for a mutation. All right, so now the way this works is very, very similar to the way that it works natively in bacteria, but the main difference is that you as a scientist are designing the sequence that you're targeting. So let's say you want to have a mutation in a very particular region of the genome. You design an RNA sequence that is complementary to that sequence, and that will then target that region for mutation. So that's called a guide RNA, sometimes referred to as an sgRNA. It's basically the same thing as the Crispr RNA that we were talking about before in bacteria. It's just going to be complementary to a specific target gene. There are lots of specific regions in the genome that can be targeted this way. Really, all you need is a little repetitive sequence called a PAM sequence, which is shown right here. There are many such regions because it's only 2 to 5 nucleotides that have to match. Basically, 2 Gs together or 2 Cs together is a PAM region. And you need this site because the Cas9 protein binds to that site. So you're generating these little guide RNAs. They're about 20 nucleotides long. And you then introduce them into the cell along with the normal Cas9 protein. So here's your guide RNA that you've generated right here, this blue strand. And it will bind to a particular region of the genome. This Cas9 protein will then bind to that site. So the guide RNA targets where the Cas9 will bind, and Cas9 then cleaves the DNA. So now remember, this is a human cell or some kind of eukaryotic cell, and we're trying to target this specific site for a mutation. So when the Cas9 protein binds to that site, it cleaves the DNA, both strands, cuts them right in half here. And what happens when you cleave DNA this way is that the eukaryotic cell wants to repair this nicked DNA. And because DNA polymerase isn't perfect in its job, it'll start to add some bases in this location, and it will usually actually try to fill this gap in with more than 1 or 2 bases. So rather than just resealing this break, 1 or 2 bases will either be added or deleted in this region. So you can see that in this next portion of the slide. So here we have this double-stranded break. And now the cell with DNA polymerase and other enzymes tries to repair this double-stranded break, and as a consequence, inserts a couple of extra nucleotides or cuts out a couple of nucleotides. In either case, you're basically causing a small insertion or a small deletion at that targeted site. Now, if that was a gene that you're interested in, imagine now you've introduced this particular mutation. When the sequence is made into RNA and then into a protein, it's going to be different, right? It's going to probably have frame shift mutation, and the protein itself will not be the same. So you generated a mutant scenario for the cell. There is one other way that you can use CRISPR-Cas to edit the genome, and this is a pretty cool way where you have a cell that has a mutation, a known mutation. Let's say you're looking to repair a mutation in the gene that causes cystic fibrosis. So you know where that mutation is, and you want to repair it. So use the same idea of generating a guide RNA, just like we did before, inserting your guide RNA into the cell. But now, in addition to inserting this guide RNA, you want to actually repair the mutation that exists in the cell. And so you incorporate along with that guide RNA a double-stranded piece of DNA that has the correct sequence. So imagine these purple nucleotides here are the correct sequence that you're hoping to replace the incorrect sequence in your original strand. So if you introduce all these things at the same time, the donor DNA, that's the wild-type DNA, the guide RNA that targets this strand of DNA, and the Cas9 protein, then what occurs at a relatively low frequency is that the DNA can actually exchange with the donor DNA can be exchanged with the original piece of DNA. So you have this double-stranded break that's been caused. You have the donor DNA with the correct sequence. And because these sequences are identical, there's something called homologous recombination, which allows this chromosome to actually pick up this strand of DNA, which contains the correct sequence, and then go back. And once it's picked up that correct sequence, then the gap is filled in to be complementary. So you've now basically repaired the mutation that was here before by replacing it with a wild-type sequence of DNA. So this is a really cool technique, obviously, that would allow you to potentially repair mutations in human cells and then reincorporate those cells into a person and potentially as a treatment. So we'll talk more about other applications for CRISPR-Cas in class.